Good day, this is the Professor, CBMD PhD, and today's Thursday, March 16, 2023. It is 10:44 and 27 seconds, p.m. Eastern Standard Time, and today's research, cellular waste. So let's jump to it here. As cellular waste removal differs according to cell type, study identifies different types of so-called lysosomes. Date March 16, 2023. Source: University of Bonn. Miniature shredders are at work in each cell, disassembling and recycling cell components that are defective or no longer required. Researchers say the exact structure of these shredders differ from cell type to cell type, a study now shows. As for example, cancer cells have a special variant that can supply them particularly effectively with building blocks for their energy metabolism. Lysosomes are a central part of the cell's waste disposal system. The tiny bubbles, surrounded by a fat-like membrane, function like a miniature recycling factory. They break down defective cell components, harmful molecules, or proteins that are no longer needed into their individual parts. And they then make these available to the cell again. The process is extremely important. If it doesn't work properly, diseases like Alzheimer's or Parkinson's can result. Lysosomes have a very complex structure. Several hundred proteins are now known to play a role in their function. There could even be significantly more when lysosomes are isolated from cells and their composition is analyzed with special equipment. Researchers often find more than 5,000 different cellular proteins. However, it is impossible to say how many of them are actually important for the lysosome's work. They can also be molecules that are in the process of being broken down in them. Others may be attracted to their membrane from the outside without performing any task, and there is usually a lot of unwanted bycatch when isolating the lysosomes, too. The researchers have developed a method that allows them to identify a large proportion of these uninvolved molecules of the 5,000 proteins typically found using conventional methods. This left around 1,000. They performed this step for 6 very different cell types, including, for instance, liver cells and cancer cells. The research explains several hundreds of these 1,000 proteins were present in almost all lysosomes, no matter which tissue they originated from. As these included about 100 new lysosomal proteins in addition to those already known. They believe it is likely that these also play an important role in the function of the nanoshredders. As what differed from cell type to cell type was the quantity in which each of these proteins was present. The lysosomes of liver cells, for example, are packed to the brim with degradation enzymes. And that makes sense. An important function of the liver is to break down different molecules. In contrast, in the cancer cells they studied, the lysosomes contained a lot of transporter proteins. As tumors require a lot of energy for their growth, at the same time, they're often poorly supplied with blood, and therefore digest the surrounding tissue and use the breakdown products to obtain energy. As digestion takes place in the lysosomes, which then have to transport the broken down molecules back into the cell, hence the many transporters. This means that these nanoshredders differ according to the requirements of the tissue in which they are found. In which of these 6 cell types they study, the lysosomes have a very specific protein makeup. Researchers point out, on the one hand, the results are particularly interesting for basic research. On the other hand, they also shed new light on the role of lysosomes in certain diseases. There are more than 1,600 studies worldwide on a wide variety of disorders, suggesting the involvement of lysosome proteins. As for example, it has been known for some time that the lysosomes, the very specific nerve cells, are altered in Parkinson's disease. We can now take a kind of protein fingerprint of these lysosomes and compare it to that of healthy individuals. And this could provide clues to how the cellular shredder function is altered in affected individuals, and why that causes neurological problems in the long term. This can also help find new approaches for drugs. And this is a Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.